Hey there. How are
Hey
you?
Shane, doing fine. How are you?
I'm good.
Okay, good, good, good. Um, yeah, so today I wanted to meet to go over some of the biology stuff, biochem stuff for my blueprint full length.
Mhm.
Um, but I didn't want to forget this, so it's going to be the first question I ask. Um, when do you think I should aim to take my next like full length exam?
Let me see. So,
Right now we have it scheduled for
yeah.
next Saturday. I can't. Well, actually, I can do next Saturday. I just realized I can do next Saturday if I want to. Do
Mhm.
you think it's worth it though? Because I spent like honestly a lot of time reviewing my Blueprint full length five.
Mhm.
Like maybe like because usually we say just a full day and then continue onwards. I don't know. I really want to make sure I got every question down, Pat. And like really understood it all. So
Mhm.
my question to you is, do I take it this Saturday?
Mhm.
In a week, or do I take it the following Thursday? Because I can't take it like that Saturday. I can't like the second.
Wait, you're saying that you're gonna take it like oh whether you're gonna take it on the second or not?
My question to you is basically,
Mhm.
do I take it on October 26th or October 31st?
I see. Um, and is that because you're not gonna I know we left like November 2nd and 3rd, like because you're doing something on those days?
Right, right, right.
Okay.
I've been like studying a lot. But though that weekend I have a couple buddies coming down, so gonna be with them. But yeah,
Got it, got it. So yeah, I think yeah, you could take it, either the 26th or the 31st. The 31st would be probably better because you'll have a lot more, you know, questions and stuff under your belt. Like, I think by that point, or you're at the point where you're doing like URL targeted stuff and section bank stuff. Yeah, you're still working on the section bank, right?
hmm. I mean, as I said, the past week it's been a little hard
Hmm.
because like I was trying to go over
Yeah.
all the, things that I don't really know well.
Hmm.
Like just my test, yeah.
Uh-huh. Yeah. So, so yeah, I think if you take it on the 31st, you'll have more questions that will have done and understood. So, yeah, it can be either of the days, but the 31st, I would say, is probably better.
Okay,
Mhm.
got it. All right. The only thing though I will say is like I won't be able to review it on the first or second,
Mhm.
but like that's not a big deal, I guess. I'll go over it on Sunday, which is like
Uh-huh.
2 days after, 3 days after.
Yep.
Okay. I'll share my screen.
Yep. Okay. So, yeah, why don't you read the passage out loud?
Hmm. Okay. In order to evaluate whether nucleic acids or proteins compose genetic material, scientists at the turn of the century employed Enterobacteriophage T2. The phage consists of a protein coat enclosing double-stranded DNA, as shown in Figure 1, and is unable to reproduce extracellularly. It can, however, reproduce within the bacteria which it infects. The phage functions by attaching to target bacteria and injecting viral DNA. Um, yeah, while the protein coat remains attached to the cell surface. After reproducing within the infected cell, the cell lyses, and the phage progeny produced using the bacterial host enzymes are released. Okay. Yeah. Step one. Scientists incubate a bacteria in media containing either phosphorus or sulfur for 4 hours, during which time the bacteria grew quickly. T2 phages were then introduced into the isotope containing media, infecting the bacteria present. After lysis, the progeny in both the 32P and 35S enriched media were found to have incorporated isotopic labels. 35S and 32P labeled phage progeny from step 2 were then introduced into media lacking isotopes. When the progeny of the resulting phage replication were analyzed, it was determined that the progeny of those phages originally labeled with 32P remained labeled, while the progeny of the phages originally labeled with 35 were unlabeled. Finally, the intact progeny from step 3 was treated with DNase, which failed to introduce any 32P into the solution. The experimenters concluded that DNA, not protein, was the genetic material involved in bacteriophage replication.
Okay, so it seems like, they're doing some experiments with radioactive phosphorus and sulfur. And I guess in step two, when they were, okay, so they started off, oh, all right, so they started off in step one, growing bacteria in radio-labeled phosphorus and sulfur. And then in step two, they added the phages. Um, and they infected the bacteria. And then after lysis, when the bacteria breaks open, the let's see, the progeny, I guess the progeny they mean like of the virus that includes the radio-labeled phosphorus and sulfur. And then in step three, they took that labeled phage progeny and then they introduced it to media lacking isotopes. And eventually it seems like the P labeled ones were still labeled, but the S ones were not labeled. So that kind of means that could mean something like that it's taking sulfur, unlabeled sulfur from like the unlabeled environment. So that could be something that they're losing or replacing, right?
What do you mean exactly by that?
So the um sulfur and the radio-labeled phosphorus and sulfur, um when you take this phage virus into and you put it into unlabeled media, it still has the labeled phosphorus, like like this is from step two, right? Um, and when they put it in media that do not have the isotopes, which are the radio labeled stuff, um, that they still have the ones that had the radio labeled phosphorus, they still have that radio labeled phosphorus, but the radio labeled sulfur like got replaced with regular sulfur,
Yeah.
right? So that must mean that something's happening in which the phage loses sulfur like to the environment because it loses its radio labeled sulfur and it seems to be taking in unlabeled sulfur from the environment.
Mm-hmm.
That does that make sense?
Yeah, I mean, not fully, but as we'll go through the questions,
Mm-hmm.
I'll ask follow-ups.
Okay. All right. Let's see. A follow-up experiment revealed that the genetic content of the bacterial cells was altered by the transfer of material from the phage. Okay.
So I got this question right. Um, I just wanted to make sure that like I kind of know all these processes.
Yeah.
Um, like so I mean the answer is transduction.
Mm-hmm.
I know transduction is when the bacteria basically, gets the genetic material from the phage. Like the phage infects the bacteria.
Hmm.
Um, I know transformation is that one I'm a little iffy about, and that's what I want to ask you about. But I guess it's when bacteria like picks up something from the environment.
Hmm.
Um, like if a cell lysed, I guess. And then conjugation is when like bacteria, it's kind of like bacterial reproduction in a way. Like the bacteria has the pilus or pilus or whatever.
Hmm.
The plus one connects to the negative pilus of the other bacteria
Hmm.
and transfers genetic material in the form of a plasmid, from what I understand.
Hmm.
And in translation is just that has nothing to do with any of that. That's just like a filler answer.
Yeah, what is it though?
Translation is when DNA gets converted into RNA,
Mm.
complementary DNA. Wait, no, no, that's sorry, that's transcription. Translation is when RNA gets converted into proteins.
Yep, yep.
Okay.
All right, cool. So yeah, transformation, is when a bacteria can pick up something from the environment that it's in. So, uh, so like for instance, in this step three of the experiment here, and if we instead of adding the phage progeny, if we added like bacteria or something, or like let's say like okay, let's say we have some bacteria here with like um I don't know some I'm just gonna say it's something called A. And if we destroy this bacteria, that A will just be around, like, leftover kind of just, around like that. And if there's another bacteria, like in that same area, it can pick up the A and, take it into the cell. So that's what transformation is. It's like just yeah, picking up something from the environment, but like typically that's going to be like after a cell lysis or something. So yeah, that's something. So that's transformation. Then conjugation, exactly what you said. With like the sex pili, and the transduction is like specifically what happens with bacteriophages. Um, and yeah, translation is mRNA into proteins.
Right. Okay.
Mhm.
During step three of the experiment, which components of the progeny most likely contain isotopes?
Yeah, I was gonna ask you this actually, like as a buzzword thing. Like when we read phosphorus and sulfur, I was gonna ask, okay, where do you think these will be present? Um, and this is kind of asking that as well. So yeah.
Okay. Well, uh, okay.
So well, first let me ask this. So like phosphorus, right? Like what type of biomolecule has that? And by biomolecule I mean like amino acid or uh fat or carbohydrate like sugar or nucleic acids like DNA, RNA. Um, which of these things? Oh yeah, yeah. We're not going to need the periodic table for this one, but yeah. So could what like a biomolecule could include phosphorus?
I think nucleic acids can include phosphorus.
Yep, 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 yep. Good. And then what about sulfur?
Sulfur, amino acids.
Yep, like cysteine and methionine, right?
Mm-hmm.
And I don't think lysine has a yeah, it doesn't have a sulfur.
Nah.
But yeah, so if they asked step two, our answer would be both of these, right?
Okay.
Because step two involved the progeny having the radio-labeled phosphorus and radio-labeled sulfur, right?
Yeah. Yeah.
So you would see the phosphorus in the double-stranded DNA, and they told us that it's double-stranded. And you'd see the sulfur in the amino acids that have sulfur in it, cysteine and methionine.
Hmm.
But since they're asking about step three, that's when it's only the phosphorus radiolabel that remains,
Right.
not the sulfur anymore.
Hmm.
So now it'll only be the DNA.
Got it.
Mhm.
I mean, C is easy to eliminate, and then like
Well, what
D
was the reason to eliminate C?
and lysine. The
Okay.
lysine doesn't even have sulfur, so
Yep,
I don't
yep.
know where they're going with that. And then D, like
Uh-huh.
um, I feel like single-stranded RNA like doesn't have anything to do with this.
Uh, yeah, just because we're radio labeling the DNA rather than the RNA. So so yeah, that would be a good way of going about it to be like, okay, um, like as you read the passage, like when you read the phosphorus and sulfur, to think, okay, phosphorus that would be in the DNA, um, or like theoretically in the RNA, but since they're doing it to this, it's DNA, so it's going to be the DNA. And then sulfur, think, okay, cysteine, methionine. So we can get rid of C, which is just kind of banking on the student forgetting which if lysine had a sulfur or not. But yeah, we would eliminate C for that reason. And then, uh, if it was step two, it would be both, A and B, but it's step three, so it's just A. And then, yeah.
Okay.
Mhm.
It was proposed that nucleic acids were composed of a single tetramer of four nitrogenous bases linked by a circular sugar phosphate backbone. How does this new information challenge the experimenter's conclusion regarding the source of genetic information? Oh yeah, I don't know. That's
Okay. So, okay, let's think about this question stem here. So, nucleic acids, a single tetramer of four nitrogenous bases linked by a circular sugar phosphate backbone, single tetramer of the four bases. Okay.
yeah, I mean like I didn't really know what that meant. Like what's GC and AT? Um, 'Cause, I mean I only am used to hearing like tetramer and like
Yeah, like in protein.
proteins.
So, um, maybe it's something like, I don't know.
That's what I thought. Yeah. Just to make sure, like DNA, right? So it's you have the phosphate bounded to the sugar ribose.
Hmm.
The phosphate attacks the 3' prime OH of the sugar.
Oh, so the um so this is like how the oh wait, sorry, it's a 3 prime.
Yeah.
So this is the 3 prime OH group, and this is why you need that, like, in order to add nucleotides to that, like you need the free 3 prime end. And what do you think happens here?
The O on the hydroxyl attacks
Yep.
the P, which is an electrophile. Well, the
Hmm.
PO, but
Yep. So that's how the polymerization happens.
got it. Okay.
Okay, so okay, single tetramer of 4 nitrogenous bases linked by a circular sugar phosphate backbone. Um, so circular sugar phosphate backbone. Okay, so if I'm imagining it to look like this, I mean, I know that AT bind and GC bind, but I mean, I don't know if I had to make like the A bind to the G or something like that.
Yeah.
But I think what we can at least say is that if it's circular, so DNA is like, the double helix, and that's like central to its function of being able to unravel and then like the two strands separating and then the so like two strands separating and then a new
Yeah.
thing can form like that during replication. Um, I don't know if that would be possible if it was like a circular thing. Um, I'm going to just look at these choices. Okay. The model does not contain sufficient structural complexity to explain genetic variation. Um, okay. The model could not versus does not. Okay. Could not explain the relatively equal ratios of nitrogenous bases found in most cells. I mean, if the single tetramer thing is making me think that it's just one thing that could be related to like relatively equal ratios, but let me see what else. Okay. Nucleic acids would not be susceptible to cleavage by DNA ACE enzymes. So the DNase enzyme, I don't know if it breaks like a double strand, like breaking like the phosphate backbone, but they both will have the circular version should still have that sugar phosphate backbone. So, you know, I don't think that it would just because it's circular that it won't be susceptible by the DNA ACE enzymes. Um, the model cannot account for the significant quantity of nuclear material present in cells. Um, not account for the significant quantity of nuclear material present in cells. Oh crap, they talked about DNA in the passage. Okay, finally, the intact progeny from step 3 were treated with DNA ACE, which failed to introduce any radiolabeled phosphorus into the solution. Then they concluded that DNA, not protein, was genetic material. Um, did not introduce any P32 into the solution. So, okay. I'm thinking that this one could not or could explain the relatively equal ratios if we have like a single tetramer. Um, or I don't see anything about okay, so I guess the way that I would see this is just that the main difference is that if it's a circular DNA, that it won't have this ability to like, you know, unravel and then, have the new DNA form like through that polymerase thing, like whatever polymerase is adding nucleotides. And I can't see that happening if it's circular. Because like I don't even know if it's like if it's one sugar phosphate backbone or two, because in DNA we have the double helix, and that's essential to its ability to replicate like this. So I think that so if I just go, okay, let me just go do this one more time. So the model cannot account for the significant quantity of nuclear material present in cells. Nothing really suggests that. Nucleic acids would not be susceptible to cleavage by DNA ACE enzymes. Nothing suggests that. The model could not explain the relatively equal ratios of nitrogenous bases found in most cells. I don't see why it wouldn't be able to do that. I think that the main thing would be probably the genetic variation, because that's when we always like genetic variation happens to us when we, uh, you know, for instance, like replicate or have some type of, uh, it could be replicate in mitosis, but it could also be like in meiosis where, you know, you have like the father DNA strand and the mother DNA strand, and they can combine um, and have like the homologous chromosomes can have crossing over where their genetic variation is being created. Um, but that's central to the fact that it's a double helix that can unravel. Um, if it was any other type of shape, uh, it shouldn't be able to do that. So if it's a circular thing, then uh I don't think that it can be. Yeah, first of all, I don't even know if it's two strands. Um, but even if it was, just if it's a circle, it can't do this. So that's what I would be probably leaning on.
This is what they said.
If nucleic acids were composed of a nucleotide tetramer of only four bases, then the significant genetic complexity arising from the enormous number of possible linear sequences um, of single nitrogenous base containing nucleotides of potentially significant length, would not be possible. I guess just enormous number of possible linear sequences, perhaps. I mean, this, this isn't very helpful, but
Yep.
I think that's the thing with the linear, breaking off into like the new types of things. So
Hmm.
I think that's the key thing.
Okay. Oh my God. Okay. So, which of these, if true, would most challenge the researcher's conclusions regarding the identity of the genetic material involved in the experiment?
Like a cars question.
Yeah, I was just thinking that.
Um, okay, yeah, what are your thoughts here?
Okay. So, identity of genetic material. Transfer of DNA between phage and bacteria did not occur immediately following insertion of the virus. Yeah, I mean, this was probably the hardest question for me. I mean, there's four, yeah. This is the hardest one. Um, I had no idea. These were all like pretty enticing.
So
So, yeah.
we might be able to do that like cars strategy here with weakener questions where you take the main idea and you
Mm-hmm.
flip it. So
Yep.
what was the researcher's conclusion?
Um, DNA was the genetic material involved.
Yep. So if we flip that, we could say protein is the genetic material.
Mm-hmm.
So maybe we can look for that.
Okay. Transfer of DNA between phage and bacteria did not occur immediately following insertion of the virus. Uh, it's not like it's a pretty broad answer. So I don't know. I'll keep that and go to B. Presence of 32P was identified in the phage progeny following the lysis of infected step 3 bacteria and consequent release of phage virions. Um, presence of P32. But there was P32, so I don't know. I feel like that wouldn't challenge it.
Yeah.
35S containing code fragments from phage progeny adsorb two bacteria, although the fragments contain no DNA. I don't know. Hmm. I mean, I don't know what that has to do with like. I mean, they say although fragments contain no DNA, but like,
Mhm.
well, I eliminated B. Between A and C, I'll eliminate. I just think A is such a broad answer.
So we know we're dealing with DNA being transferred between, from the phage to the bacteria. Um, what's so in for A, what there's like a key word in there that makes this makes it more than just saying that that DNA is being transferred.
Um, Immediately.
Hmm. Good. So we were able to, or the researchers were able to reach their conclusion by letting phages transfer DNA to bacteria. Now, if it just happened, or if it didn't happen immediately, would that change, you know, the conclusion? I guess, it shouldn't, right? It just means that it would take a little bit longer, but you're still having DNA being transferred from the phage to the bacteria. Um, and, it shouldn't matter if it's fast or not fast, right?
Okay. Got it. Yeah.
So, so yeah, the immediately part shouldn't matter.
Hmm.
And then the presence of P32 was identified in the phage progeny following the lysis of infected step 3 bacteria and consequent release of phage virions. Um, yeah, okay. Yeah, so we know that that's what happens in step 3, right? Um, so that should support,
Hmm.
okay, so that's out.
Yeah.
And then uh S containing coat fragments from phage progeny adsorb into um bacteria, although the fragments contain no DNA. What are your thoughts here?
So, uh, I don't know what this has to do with like, I mean, they say the fragments contain no DNA, but I just think D, I guess, does challenge it more because they say protein entered from the phage, you know?
So uh let's see. So for C, the S coating containing coat fragments um adsorb to bacteria. Uh and the fragments contain no DNA. So we know that they shouldn't contain DNA because DNA doesn't have sulfur. So this is just saying that bacteria can absorb um, fragments of the phage's coat that contains that radiolabeled sulfur. And that is just the coat, right? And we know that in the passage, if you go maybe up on the left, that, yeah, this is the protein coat. So, like, yeah, when this breaks apart, pieces of that protein coat, let's say this is like a piece, and this is your bacteria, it could take it in, right? But it's not taking in any genetic material here. Um, it's just taking in like the proteins. And so
Okay.
that is something that happens. And so so that one should be out as well. And then D, protein which happened not to pick up the S35 label entered the cell from the phage. Okay. Now this is saying that this thing, whatever contains that, um, so this would mean that the phage, instead of because it normally would inject its DNA into the bacteria, this one is saying that it's injecting, some type of thing with sulfur, which must be a protein, into the bacteria. So if we're saying that the proteins, the S labeled stuff, are, instead of coming from the environment, if they're coming from the phage, then that would challenge that researcher's conclusion of the DNA being the thing that can transfer from the phage to the bacteria.
Okay.
Like, so basically this entire experiment, right? It's about, for instance, starting off with bacteria and and incubating it in, radiolabeled phosphorus and radiolabeled sulfur, and then phages were introduced, and they, you know, infect the bacteria and all that stuff, and take advantage of its machinery to make more of itself, which to make more DNA of itself, which means it's going to incorporate the phosphorus, the radiolabeled phosphorus, but also to make the proteins, I imagine, for the protein coat, which would be which would take up the radiolabeled sulfur. So that's step two, like what that's showing. And then step three shows us that when you take that progeny and you put it into, media that lacks the isotopes, the original label stays for the phosphorus, aka DNA, but the one for the protein does not. So basically, we get to say that, okay, it's DNA because uh when you start off with the bacteria having the labeled phosphorus and sulfur, and then you see that the phages start to have that too. So that means that there's and particularly the phosphorus one, because that one is remains labeled regardless of the surrounding environment, which means that that label must have come from that, initial phage, entering inserting its DNA into the bacteria. So I think that would be the one that most challenges it, um, because you get to see that, like, because if you think about it, I guess DNA is the thing that remains intact through generations. And so the intact progeny from step three, like, it, it incorporates the P label. So it means that, that supports the conclusion of genetic material being, DNA. And the S label would be, if it was the case, it would mean that the phage is inserting some protein rather than DNA into the bacteria.
So just so I understand the isotope, the reason they like, what's the point of using an isotope here? Is
Oh,
it because it's used as like a label,
yeah,
and so they're able to see through their experiment,
yep,
like they're able to track the DNA
yep,
versus protein. That's why.
yeah, exactly. Yeah. Radioactive, or, also known as radio-labeled isotopes. Yeah, so that's how we know things like a mechanism, right? Like the thing that I always draw,
Yep.
the OH, attacking, here or whatever. You could like there's no way for us to know that. I mean, we can say like, okay, like let's say this is a leaving group and this turns into a carboxylic acid. We we could say, okay, if I take an alcohol and I take a something with carbonyl in it, um, and we mix it together, then this would turn into this over here, and that's what we get as a product. But And we can do that, in a lab and stuff like that by just mixing these things together. But there's no way for us to know, like, specifically, like, which atom attacks which, and, the specific stuff like that. And radio labeling things is how we can figure out mechanisms like that. So, yeah. So like, I would imagine, like, step one being, a medium, like, full of radio labeled phosphorus and radio-labeled sulfur, and they have the bacteria in it, and that should the bacteria should incorporate the radiolabeled phosphorus and sulfur for the bacteria, and then they introduce the phage, which inserts its DNA inside and uses the bacterial cells replication machinery, uh, transcription machinery, translation machinery to make uh, more of its DNA, so it can make more, viruses, more uh, proteins to make that outer, cap or whatever. Um, and so if we see that the phage, so like, let's say this cell lyses like that, and we have a bunch of phages, and and by the way, this media still had the P and the S, but this no longer does here. This is step three. And we see, oh, you know what? The phages, they all have that radiolabeled phosphorus. So that means that, and then what carries phosphorus? DNA. So we could see that DNA is what's passed from this early like generation here into the progeny. But what we did not see in the progeny at step three was the S label. That did not make it through.
Hmm.
So if it happened to like not pick up this S label, um, that entered the cell from the phage, if the phage is transmitting uh that S label into the bacteria, what has the S? Like proteins. So if that was the case, then we could say protein is the thing that is transferred, from that first parent generation to the progeny generation. But we didn't see that. We saw that the sulfur became unlabeled. So that's not something that, is actually coming in and being passed down. But since the phosphorus remains labeled, that must be the thing that gets passed from, one generation to the next. And yeah. And so D, if that was true, that would be the most um would challenge that hypothesis the most.
Okay, got it.
Mhm.
Give me 1 minute.
Sure.
Okay.
Mhm.
All right, so the tumor suppressor P53, originally named for its apparent molecular mass, is referred to as the guardian of the genome for the many roles it plays in preserving genetic stability. While the complete picture of the protein's activity is not fully understood, to date, P53 has been identified to recruit, even initiate, identify recruit with DNA repair proteins to the site of DNA damage. Um,
Wait.
yeah.
Oh, sorry. There's we need to. Sorry. All right. Yep.
Um, yeah, okay. Recruit DNA repair proteins to the site of DNA damage, arrest cell growth in the presence of DNA damage, and even there we go, even initiate apoptosis if the DNA damage is found to be irreparable. Given the critical role p53 plays in these regulatory pathways, it should be unsurprising that the tumor suppressor gene plays a significant role in cancer development and progression. Genotype studies have confirmed that p53 has been mutated in the progression to cancer in over 50% of tumors, making it one of the most important clinical targets for gene therapy-based cancer treatments. Given p53's pivotal role in cancer development, a researcher set out to study whether transduction of wild-type p53 into cancerous cell lines could arrest cell growth. To this end, three colorectal cancer cell lines, as well as a benign tumor cell, PT135, were selected 134. One selected for the study in order to assay for growth suppression phenotype. The researchers constructed two plasmids, one that contained wild-type copy p53 gene, and another that contained a non-functional missense mutated copy of the p53 gene. Both plasmids were otherwise identical. And for selection purposes, the constructs were engineered to include the mammalian neomycin resistance gene, which conveys resistance to antibiotic gentamicin. To assay whether p53 transduction could suppress cancerous growth, cells from the three colorectal cancer cell lines and the benign tumor line were altered with PC2753, PC2753X, respectively, to select for successfully altered cells. The cell lines were cultured in a solution containing 0.8 milligrams per milliliter of the antibiotic gentamicin and left to incubate at a temperature of 37 Celsius. After 34 weeks, the number of colonies in each culture were counted. Results were tabulated to form table 1.
All right. So let's take a look at the figure one. So what do we have there?
So we have the four cell lines.
Oh, I mean for figure
Oh,
one.
sorry.
Mhm.
Figure 1 just shows the plasmids. The one on the right is the mutation. It has a missense mutation, and yeah.
So um so they talked about the different cell lines. So yeah, so we talked about these cancer cell lines and then the benign tumor cell line. And I think that the benign tumor cell line might be used as some type of control, perhaps,
hmm
because could be like some two-step hypothesis thing. But then when we have the plasmids, one of them works and the other one does not. The one with the X does not work. Okay, so doesn't seem to be anything else that's very different between those. Okay, let's look at that table. Okay, the 157 line is, let's see, 58, and then for the mutants, okay. Transduction, okay. So CRC 157, like way more of the X. 169, similar amounts. 184, Um, a lot more X than wild type, one, and then the 134, the B one is like similar amounts as well, but significantly smaller.
Mhm.
So, okay. Um, whether transduction could suppress cancerous growth. Okay. And then, okay. All right. So let's see. If the neomycin resistance gene on 53 and 53X plasmids was abnormally susceptible to mutation, the most likely effect on the results from experiment one would be that, um, okay, so I guess, so yeah, tell me about your approach here.
So basically I thought that well neomycin, they talk about the neomycin resistance here.
Mhm,
Conveys resistance to the antibiotic gentamicin.
mhm.
So if it's mutated, I thought that they would not be resistant to the antibiotic. And so
Mhm.
I thought they would decrease in amount.
Okay. Let's see. A, the number of colonies observed only for CRC 157 and CRC 184. Um, 157, 184. So the number of colonies observed only for that and that with the wild type would decrease. I don't know why 169 wouldn't. Um, so I'm iffy on that. There would be a decrease in the number of colonies in all trials. I guess that could work. Um, okay. Number of colonies observed only for 169 transfected with 53 and 53X would increase. So that's talking about the 169, but it shouldn't increase, right? Because you're messing up, you're making it no longer like I'm imagining it being something like that makes you not resistant to the antibiotic, right?
Yeah.
So that's the opposite. And this one, A, I don't know why they wouldn't include the other ones. And then D would be definitely the opposite. So I think it should be B.
Yeah, it is. Okay. I mean, I understand it now actually after doing it the second time.
Yeah, what I don't like about this is this mutation. I mean, like they already have an example of a missense mutation. Like that doesn't change anything, right? There could there could be like in order for us to answer this question this way, it's we have to infer that when they say mutation, they mean a mutation that messes it up.
Yeah.
So yeah.
Genotypic analysis of another colorectal cancer cell line CRC200 indicates that the cancerous cells are expressing a single nonsense mutation in one of their copies of P53. Given that information, which of the following would most likely represent the number of colonies observed after a transfection assay identical to the one used in experiment 1?
Tell me about a nonsense mutation.
Nonsense, it's a mutation to a stop codon, so it stops something prematurely.
Yep.
Let's go through all of them, honestly. Missense mutation. Um, that one I kind of forgot about. Is it when you change like one of the you change one of the parts of the codon so it completely changes?
It just code. It's like it'll code for a different amino acid, um, which could like not matter. Like if you have like serine turn into threonine or something like that. Um, but it could also matter if the amino acid is completely different from the one that it replaces. But yeah, a missense mutation is just you know um whatever amino acid you're supposed to get for a codon, you get something else.
Okay.
So yeah, so there's the missense one, which is just it's the wrong amino acid. And then the nonsense one is when you have that premature stop codon and it stops that from being translated and that really messes stuff up. And then you have silent mutation. Oh yeah. So silent mutation is when you have a missense mutation that does not. Oh wait.
I get it. I get it.
No,
Nothing
no, no.
changes.
Sorry.
I know what you mean.
Yeah. Silent does not change that amino acid. So okay, so
Yep.
these are the ones, and then so okay. In the copies of P53 have the nonsense mutations for CRC200. And if you did the assay like in experiment 1 to that cell line, um, what what effect would that have on the amount of all this stuff? So, okay, let's see. 53, 6, and then the other one 56, and then 67 and 713, 712. Um, okay, so okay, so I'm going to compare it to the wild type. So this one, I'm going to use a down arrow to mean it's smaller than the wild type and up arrow to be if it's larger. And then the 56 also smaller. And then for the, oh wait,
This is just like wild type and mutants.
yeah, I was looking at the different values for the chart from the table, but it's not specifying any of those particular ones. Um, so let me think here. So, okay, P53, tumor suppressor gene. Okay. Okay, wait. Single nonsense mutation in one of their copies of P53. One of their copies. So there should be another copy, right?
Okay.
Just because it's a human cell.
Yeah.
So, and we know that P53, is something that suppresses cancerous growth. So I would imagine that the number of the resistant colonies would be very, very large across the board. I think that's D then.
Yeah.
Because okay.
So if something has this, everything has two copies of like every cell, every I mean um eukaryote, I guess, has two copies of a gene. One
Uh
from mom, one from dad.
so like for a human, for instance, yeah, because our body cells are diploid, containing two sets, and our sex cells are haploid, containing one set. So since this is a human cell from, colorectal cancer, uh, and they say that there's a single nonsense mutation in one of their copies of P53, that must imply that their other copy does not have a mutation, right? Because they would have said it, right?
Yeah.
So, um, but yeah, for the most part, um, if you are, I guess, damaging that P53 function, yeah.
Mhm. Okay. Next is this.
Which of the following statements describe an obstacle researchers would face in implementing a similar transduction procedure to treat cancerous growth within patients? Um, okay. Uh, Gene incompatibility. So, okay, let me think about it. So, researchers are like, okay, let's treat cancerous growth using transduction. Uh, what could be an obstacle to that, right? So incompatibility between patients with different endogenous uh copies of P53. Um Different copies of it. I don't think that would be lead to incompatibility, but okay, let's see. Roman numeral two, patient immunological response to the that's a good one. Okay. So tell me again, what's transduction?
Transduction is when you have a virus that injects its DNA or RNA
Hmm.
into the bacteria.
Hmm. So what do you think about Roman numeral two?
Uh, I feel like there could be a response, yeah.
Yeah, right. Because we're doing that with viruses, right? And so if you want to treat cancerous growth within patients, give them a virus. The virus, isn't gonna like negatively affect them, but the immune system is gonna be like, what's this? I gotta, kill it.
Yeah.
So that can happen. Effective virus delivery into the entirety of the tumor. Okay. So what is that saying?
That it can like fully get the virus to kill the whole tumor, I guess, or affect the whole tumor.
Yeah, or like deliver, so like, yeah, deliver the virus to the entire tumor, which means delivering it to all of the cells, the cancerous cells that comprise the tumor, right?
Mm-hmm.
Um, which, you know, could be an obstacle, right? So I think then I don't see how a gene incompatibility would happen if you have different copies of P53. Um, like considering the fact that we can literally, insert it into plasmids, like these plasmids in bacteria have this human gene in it. So if that's like that shows that, it can be completely foreign P53 that we add to like a bacteria. There's no issues with gene incompatibility there. So, why should there be one between people who are like humans and P53 is a human gene? So, yeah, so I don't think one would be likely to happen.
Okay. Yeah, I knew two was right, but I wasn't sure that three was right or not. So,
Yeah, so this one
okay.
I just take to mean entirety of the tumor, all of the tumor cells, right?
Mm-hmm.
Mhm.
Oh wow, that's it. Okay. Um, yeah, so there was just one other passage that I want to go through. It was like from a long, long time ago.
Okay.
One second.
Mhm.
Do you see it?
Mhm.
Okay. So when present in the bloodstream above a threshold partial pressure, general anesthetic blocks the sensation of pain, induces a state of unconsciousness, and causes muscle paralysis. Many anesthetic agents are administered in the gaseous phase and are absorbed into the bloodstream through the lungs. The rate of diffusion of an anesthetic across the alveolar membrane with a surface area A and a thickness T is described by the Fick law of diffusion, where D is the diffusion coefficient specific for the gas and delta P is the partial pressure difference across the membrane. An inhaled anesthetic is described by its blood gas partition coefficient, the ratio of the anesthetic's concentration in the blood to its concentration in lungs when the partial pressure is equal. Concentration C of anesthetic gas dissolved in blood is described by the Henry law of solubility, where KH is the solubility constant specific for the gas and P gas is the partial pressure of the gas. Due to the paralytic effects of general anesthetics, mechanical ventilators are usually necessary to assist the patient's breathing during surgery. Positive pressure ventilators are non-invasive and use external pumps to induce inspiration by forcing air into the lungs through a ventilation mask. Despite the paralytic effects of general anesthesia, patients are often capable of expiration with the active aid of a mechanical ventilator.
All right. So, um, the anesthetic agents administered like through gas, and therefore they get absorbed into the bloodstream through the lungs. The rate of diffusion across an alveolar membrane with a surface area A and thickness T is this Fick law of diffusion thing that we have.
Mhm.
Um, D, okay, diffusion coefficient, and okay, an inhaled anesthetic blood gas partition coefficient ratio of the anesthetic's concentration in the blood relative to its concentration in the lungs when the partial pressures are equal. Concentration C of an anesthetic gas dissolved in blood is Henry law of solubility. So we have that there, constant, and then pressure, partial pressure of the gas. Um, Paralytic effects usually need mechanical ventilator. Positive pressure ventilators, non-invasive, and use external pumps to induce inspiration by forcing air into the lungs through a ventilation mask. Um, Despite the paralytic effects, often capable of expiration, like exhaling, I guess, but not the inhalation part. So okay. Which of the following graphs best depicts the anesthetic partial pressure along the length of the pulmonary capillaries where it is being administered? Capillaries are in contact with alveolar membranes along the entire length shown in the graphs. Assume there is no anesthetic in the blood when it enters the lungs. Okay. So walk me through this.
Yeah, I had no idea. I thought it was B, but it wasn't. Um,
Uh-huh.
yeah, I have no idea. I mean, I just thought that it was like my thinking was like as you enter the capillary, like as you're getting to it, the vessels get smaller and smaller and smaller. And then they start getting bigger, bigger, bigger again. Just like arteries to arterioles to capillaries,
Mm-hmm.
venules and veins. That's why I thought it was B, but
So
oh
it's
yeah.
showing that the partial pressure reaches a max, like goes up, reaches a max, and then goes down. Um, but in a capillary, you know, um, when you have like the red blood cells in single file and gas exchange happens, so it would be like, you know, O2, like from oxygenated blood into the, or rather, sorry. So, so it can give O2 from the oxygenated blood to the tissue that's undergoing cellular respiration. So it needs more O2, and it also produces an excess of carbon dioxide, which would start to enter into the red blood cell here. Um, and yeah, that should be. Oh, and so okay, of partial pressure of the is it the anesthetic, I guess, the gas anesthetic. Um, so okay, so what I wrote was like cell, cell respiration in which you have something going in, something coming out. Um, and the reason that the CO2 is coming in is because the PCO2 like is higher, in the tissue compared to inside of the blood, and vice versa. The PO2 is higher in the blood, which is why it, you know, pushes it out into the tissue. So if they're talking about the, oh wait, they're saying no anesthetic. Oh no, no anesthetic in the blood. But I think they're talking about, yeah, anesthetic gas that gets taken up by the capillaries of the alveoli. And that should mean that we should have um, an increase in the partial pressure of that anesthetic. So it gets forced into the bloodstream. Um, so, this shows that it reaches some max point, but then it starts to drop again. Um, I mean, it makes sense that it would reach a max point, but it doesn't make sense that it would drop again because if it drops again, that partial pressure is smaller of the anesthetic, and that means that you're going to absorb less of that um, anesthetic. And, they didn't say anything like that's how it happens because, if it drops like this, it would push that gas outside, like out of the capillary, right?
Mhm.
So, um, so yeah, I would say B wouldn't work, and D looks like it wouldn't work because it's just going down. And now between A and C, A shows that the slope here is increasing, right? But we know that it can't do that, forever. Like at a certain point, it's going to reach a maximum amount and plateau. So C would be more so what you should be able to see. Hmm.
So my question is like they're saying capillaries. So the anesthetic is not in the blood when it enters the lungs. So what does that even mean?
That means that, well, so if the anesthetic, you know, partial pressure of it is high compared to within the capillary, it's low, it should, through diffusion, go from high to low, right? So it should enter into the blood.
But then where's it entering from?
Oh, so it, so, um,
Like is the tissue the lungs? Like
oh,
the
I see what you're asking. Okay, so let me make this a little better. So, at the lungs, there could be a high. So, like for gases, concentration and partial pressures are going to be interchangeable in a lot of cases. So, I'm going to write like high concentration of the anesthetic, but I'm going to put a little p in front of it to make it a partial pressure of that anesthetic. Let's just call it A.
Mhm.
Um, and that's the lungs, and then this is a capillary. Um, and inside of it has not just low, but no, or I'll say low partial pressure of the anesthetic. And diffusion then would dictate that it goes from high to low. So that's the anesthetic entering into your bloodstream after you inhale it at the lungs.
Okay.
And then let's say, when it's around like a tissue, right? You have high partial pressure of anesthetic and low in the tissue. So through diffusion, that anesthetic reaches the tissue. And it just continues to do that for your entire body. So now them saying there's no anesthetic in the blood when it enters the lungs, meaning there's no anesthetic to begin with, right, in the blood. Because if there was, you know, these graphs wouldn't start at such a low value. D starts with a high value, which indicates that there's already, you know, anesthetic in the blood. And so, by that point it starts to drop off. But yeah, that's what they mean. So what's happening here is like basically the gas you inhale and goes into the capillaries of your alveoli of your lungs, which means it enters into the blood. Um, Enters into the blood because there's a higher partial pressure of that anesthetic in the lungs, so it diffuses into the blood, and then your blood, transports it throughout your body to tissues that have a low amount of that anesthetic. So this time the blood has a high amount of the anesthetic, and it can diffuse that into the tissue, and that's how you would get that. So yeah, so that's what they mean by that. Yeah, here we go. So you see how the passive diffusion.
So as you're breathing in more anesthetic, like then it's gonna start flowing into the blood.
Mhm.
Hmm. Okay. You said partial pressure and concentration is pretty equal.
Mhm.
Okay. A positive pressure mechanical ventilator most likely inflates the lungs bidirectly.
Yeah, I remember hating this intrapleural pressure thing, but um, what is that?
So basically like it's the pressure inside of your lungs. Um, there's like the extrapleural and intrapleural.
Mhm.
Uh, not fully 100% sure, but like
Mhm.
yeah.
Okay. Um, so intrapleural. Okay, so what is meant by positive pressure or, negative pressure?
So basically like your lung inflates when the pressure outside is higher than the inside. And then
Oh, I mean, so
because
which one is positive pressure? Which one's negative?
that's a thing, I don't really know.
Hmm.
Yeah. Um, I would guess
So
positive pressure is when the outside it's greater.
yeah, yeah, yeah. Good, good. Because yeah, it's a little misleading because pressure can't be negative or positive, but relative to the surroundings, right?
Yeah.
Like, you know, at the lungs here, there's this positive pressure because there's a high partial pressure of the anesthetic and a low partial pressure of the anesthetic, and it can, uh, work that way. Um, and then they said that the positive pressure ventilators use pumps to induce inspiration by forcing air into the lungs through a ventilation mask. What? And so, okay, so it's saying that it forces air into the lungs. That's your positive pressure, right?
Yeah.
Whereas, like, you know, yeah, like high pressure to low pressure, high concentration to low concentration, diffusion, all of that involves negative pressure. Um, but oh, sorry, positive pressure, my bad. But if it's something like sucking something, it's going to be the negative pressure. So, all right, so positive pressure inflates the lungs by directly intrapleural pressure. Okay, so intrapleural pressure. So, like, okay, the lungs are surrounded by that pleural space, um, which is like kind of like surrounds the lungs, and you know, I guess it equilibrates the pressure changes in the lungs when we uh oh Hmm. Diaphragm contraction increases the um, volume, uh, which would decrease the pressure of the lungs. And that happens during inhalation when you have a higher partial pressure of things outside compared to the inside, like inside of your lungs. So you can inspire that way. Um, and then expiration when you exhale, your diaphragm relaxes and it goes back up, and the overall volume of that intrapleural cavity is larger and um, sorry, smaller volume, which means a higher pressure. So that's why you exhale there. So if
So hold
they,
on, I'm gonna interrupt you real quick, just so I'm understanding this correctly.
yep,
Um, w when you're inspiring, you increase the volume in your lung, and that leads to a decreased pressure.
hmm
And the opposite is true for expiring.
hmm
Is there like, does that have any relation to do with like, not directly, but like Boyle's law, like PV equals PV or no?
yeah. So
Kind of.
that's what
Okay.
uh, mostly in terms of just the inverse proportion
Yeah.
between P and V.
Yeah.
So, uh, so basically, inhalation involves a higher partial pressure or concentration of air, like outside of our lungs, um, which is why we can inhale stuff, and exhale is the opposite. So you're changing the volume of the lungs or the pleural, whatever, pleural cavity. And when you change the volume, you change the pressure as well, and then that's going to be relative to the atmosphere. Um, Therefore, inhalation and exhalation. Um, Now, if I look at the pressure here, I mean, they have pleural pressure here, and then they have intrapleural pressure here. I don't know why it's
Probably. Yeah, I don't know.
pleural pressure.
Probably it's probably a mistake because in
Okay.
the bottom one it's intraflow.
Yep. So we have um, a negative something, then a less negative something here. So it seems like the intrapleural pressure possibly could be smaller, uh, but okay. Yeah. So yeah, let's go back to that question. All right. So inflates positive pressure inflates the lungs bidirectly. Um, so okay, if we're inflating the lungs, we are increasing the volume, which means, let's see. Okay, so um, one sec. Uh, Okay, so here, check out this image here. So, okay, so now we have a better kind of idea of the difference here. So, because like when I was thinking of alveolar pressure, I was thinking like literally on the level of like the, grape-like things, but I think this simplifies it. So we see that this surrounding like you know, gray area or whatever is the intrapleural pressure. Um, but the within it is the alveolar pressure. Um, and so at the one on the left, it's when you finish, exhaling. Um, and you can see that the when you exhale, the since we want the pressure to be um higher inside of the lungs to exhale, uh we should have the volume go like decrease inside. So that's why you see the alveolar pressure, like with those white arrows are going inwards. Um, but we also see the intrapleural pressure, you know, going in the opposite direction, like expanding outward. And, that's just because that overall volume of this, thing has like the lungs inside of the intrapleural, the pleural cavity or whatever. Uh, just so you don't have like, because like let's say that you got punctured or something here, like you would have a collapse, like a collapsed lung.
Okay.
Like if you puncture this pleural cavity, then because right now the intrapleural pressure is what lets it be the way that it is. Whereas if you didn't have it or if you punctured it, you would have atmospheric pressure pushing down on it, which is much, much greater, and you can have a collapsed lung. So that, yeah, so this entire pleural, cavity, it's kind of like protects it and also gives it like an opposite kind of force. Like so if the alveolar pressure is going down, sorry, uh the alveolar area is getting compacted with a smaller volume. To make up for that, the intrapleural pressure would go expand outward because you want that overall lung cavity to not change in volume, right? Uh, so what we do want to change in volume is the alveolar pressure or the lungs itself. So then we see that during inspiration here, we see that, well, I guess here we are having this like white arrow as the forced inspiration, like if you're using a ventilator of some sort. And it seems like, you know, so we still have these arrows going inwards, but the values go from this to this. So the alveolar pressure seems to decrease, and the intrapleural pressure, well, that also seems to decrease. Hmm. So I mean, okay, we could at least say that, or at least eliminate transmural pressure plus that plus 7. Okay, so I'm really just between decreasing um inflates the lungs. by that image that I sent should have, wait a sec. All right, the one that I sent, like, I don't know what, wait, so if this is coming in, this should expand, right?
That's why I was confused. I mean, it does show that it's expanding, but it says alveolar pressure goes down.
Yeah. That shouldn't be the case. I mean, it should expand. Um, and the pressure should increase.
Yeah, you need an editor to make your own chain customized graphics. These internet ones are not cutting it.
Oh yeah. Yeah, if you know anyone, like, I'm
Yeah.
also looking for like an editor who can make, make
Yep.
these like clickbaity kind of things with all the
Yep.
shorts and stuff.
We're
But
getting close to a K subs, so.
yeah. I need to get uh like a certain number of watch hours to like be able to like monetize it. And
Mm,
so I need like 700 more.
got it.
But yeah. So yeah, it's weird, but forced
Okay.
respiration should be like forcing stuff, into your lungs, causing an expansion. Um,
Yeah, okay. Well, now in terms of the problem,
yep.
so I just like was also really confused about what alveolar pressure was myself because I've only heard of intrapleural and
Mhm,
I'm extrapleural.
mhm,
So like when I was doing the problem, I just like didn't even look at that.
mhm,
I put decreasing intrapleural. Okay, it was wrong. So I put decreasing intrapleural because I thought you have to decrease the pressure in the lungs because like you want it to the differential pressure differential to be like higher outside. And when it's higher outside, you it stimulates inhaling.
mhm,
So you inhale because the pressure is like lower on inside.
mhm.
So that's why I thought it was B.
Yeah.
So
So I think what happens is, yeah, so we have the intrapleural space is just what keeps this entire like size of, the lungs, regardless of, I mean, so like if so like over here, I guess where the diaphragm contracts, right? We have that increased volume of that lung, and the intrapleural space should like stabilize it. And then when we have, the exhalation, when your diaphragm, relaxes, you have that smaller, or sorry, that large, a smaller volume, um, which means a higher pressure that you can exhale. So the intrapleural space just does the opposite of the what the lungs do.
look,
Keep it all. Yeah, there you go.
it's tricky.
IPE pushes
It's
against
tricky.
it's what?
It's very tricky. Look,
Hmm.
they say the diaphragm is paralyzed because the anesthetic,
Hmm.
and that's why this has nothing to do with because my reasoning was correct, I'm pretty sure. Look, they say intrapleural pressure decreases when the diaphragm contracts, but because the person is under anesthetic, the diaphragm can't be controlled.
Okay.
And that's why it wasn't B.
Hmm.
Um, but what exactly is alveolar pressure? Like, I know it's the alveoli, but
Yeah, I think you can think of it as a stand-in for the lung pressure. So yeah, so I think um that yeah, so the outer part is the intrapleural space, the inner part is the lungs, and the language that they use to describe the lungs is alveolar pressure. Um, and so we can just think of the intrapleural pressure to do the opposite, but ultimately, you know, in this diagram over here on the top right, we have the forced air coming in to the lungs. So that should affect the alveolar pressure because that's where we are adding, you know, oxygen and air and stuff.
So increased alveolar pressure basically means lungs inflating.
Uh, you mean oh, you're saying means
Lungs inflate, yeah.
mhm.
Okay. Yeah, next is this.
Unassisted expert. Okay, I had a feeling they were gonna ask this. So, um, okay, so like we know that. Okay, let's see. So you can force inspiration, um, because that isn't something that you could do, when you have anesthetic, but even with that effect of the anesthetic, people are still able to expire or, exhale without needing the mechanical ventilator. So, okay, the only one that would make sense should be this one because, and this is a good thing to talk about, elastic. Um, what do you think of when you think of elastic?
It's a pretty broad question, honestly.
Mhm.
Elasticity. I think I've heard it somewhere in physics, maybe. I don't know. I
Huh? So, for instance, like let's say like an elastic waistband, or elastic or a rubber band, right?
Yep.
So what about those things, what property, I guess, do those things have?
Flexible.
Okay. So now that's like what most people would say elastic means, but what it really means is that if you stretch it or something like that and then let go, it'll return back to that original, you know, length, right?
Mm, right.
So, because there's so basically like there's stress and there's strain, right? The stress is something that you apply to something, um, and the strain is how much that thing gets affected by it. And you know, you could have like, I think it's like the modulus or something like that, where it's like something like this, like with as strain goes up, sorry, as stress goes up, strain goes up. Um, but there could be situations where the stress and the strain are not equal, and the strain becomes a lot larger than the stress.
Mhm.
Um, so like, okay, for the elasticity part of it, I'll explain it to you like in two ways. One way with like the theory, and then the other way with eliminating the other choices. So, you know the Titanic submersible guy?
No.
So like when these like it was like maybe 2 years ago, this guy made a submersible and he invited a bunch of people on it, and then they went down to try to see the
Oh, I've heard of it.
yeah, to see the wreck of the um Titanic, and well, they imploded. Now, why did that happen? Well, two reasons, but one reason is what's relevant. So a metal is, you know how they in high school they would define it as like a sea of electrons.
Yeah.
So like imagine like what I imagine is like a big jar of like M&Ms, and if I put my hand inside of it and my hand is the metal and the M&Ms around it are the electrons, I can bend and, move my hand in a lots of different ways and the electrons will just move and let me bend.
Hmm.
For something that isn't a metal though, um it can't move. It doesn't have that mobile, electrons. So if you just bend something, right, it will cause like the electrons to repel each other and therefore it'll crack. And so typically people use metal for submersibles because they can stretch or get compressed, but come back to that original like length. Whereas something like a carbon fiber can be stretched, but it doesn't have the elastic capability, which means that it can come back to the original shape from when it's stretched. So for this question, uh, we see that to cause inspiration, we need the positive pressure the ventilators. But when your lungs are, you know, expanded, right? Uh, to allow that inspiration to happen, like they're not going to stay in that expanded state. So they're going to go back to that original state and kind of compress back down. And that's the elasticity aspect of it. Because elasticity, we think of it as things that can stretch, but it's really something that can stretch and go and return to its original, form.
So would you say metals are elastic?
Yeah, yeah.
Yeah.
So, and by that I meaning, so yeah, elastic recoil, right? So when stretched, these fibers exert a restorative force on the lungs following inspiration.
So why is D wrong?
Okay, so now why are the other ones wrong? So yeah, D is wrong because, well, I mean, we're not really talking about let's see, energy stored in the energy shouldn't be stored in the diaphragm, because that's just like a little muscle, um that doesn't have that because like in order for this to happen, like you need that elasticity to go back to stretch back to the original position. But energy stored in the diaphragm, that the diaphragm doesn't have any elastic stuff that we can think of. And the energy stored here, they're saying that it's a muscle, that it's not made up of elastic fibers, and that it simply relaxes during expiration. So that's just contraction and relaxation, as as a protein does, or sorry, as a muscle does. um but the yeah, elastic recoil and surface tension effects refers to the tendency of the lungs to return to their original shape after being stretched during inhalation. That's exactly that, the elastic factor there. So yeah, it's not going to be the Venturi effect, because that is when you have a, let's say you have some type of gas or air or fluid, and it goes really fast through like some type of tube, and let's say that tube is like open to the atmosphere or something, it'll suck in stuff because of that moving gas. So that's the Venturi effect. And then turbulent flow is when you have a real, yeah. So you
Yeah.
see that on the left, it's laminar flow. We have
Hmm.
the arrows, but turbulent flow is when it makes those eddies, those whirlpools.
Yeah.
Yeah.
Okay. This is the last one.
Okay.
Um,
So
yeah.
many inhalational anesthetics are taught to increase surface tension effects in the alveoli. When water has a slightly greater surface tension, it oh, okay, I remember that. This from like middle school or something with the meniscus. Um, didn't you tear something like that?
I did. I mean, that's
How's that
the
go?
that's a different type of meniscus, but
Yeah, yeah.
I did in fact tear it. Yep, yep, yep.
Is it like healed?
Not fully, but I've been doing physical therapy.
Got it.
Yep, yep.
Oh, all right. Convex meniscus. Okay, that's something like do you remember this from like I don't know if they taught it, but I
Yeah.
like when you're a kid or something. So like the one on the left is a concave meniscus, and that's when the adhesive properties between this fluid and the walls of the container, the adhesive part is stronger than the cohesive part. Cohesive is when it's more so bonded to itself.
Okay.
The
Yeah, yeah.
the convex one is more so the you know cohesive properties. I don't see anything like why that would happen because like surface tension isn't going to change the cohesive or adhesive properties. Let's see, evaporates more quickly. Okay, so let's think a little bit about what surface tension is. What would you say that the surface tension is? Like,
I mean, I remember very well in high school, one of the examples was the fact that bugs can just walk on water because it has such
mhm,
it's because of the polar bonds of water, and that makes it pretty strong. Like,
uh-huh. Yeah, yeah, exactly. So, yeah, so the water molecules can hydrogen bond with one another. Um, and particularly if they are at the surface, lots and lots of those bonds, like a thick layer of, water molecule bonds, that will give it a certain surface tension. So what happens in that case? Let's see, evaporates more quickly. So that would be like the opposite of what happens because in like water, it's like, let's say this is a water molecule and it's like bound to like this one, bound to this one, like this. If you have a high surface tension, you have a lot of at the surface of that water, strong intermolecular forces there, which means that they're not going to want to break out of that to enter into the gas phase or the vapor phase. So they shouldn't evaporate more quickly. It should be less quickly because like this water molecule is so attracted to this one over here that it's not going to want to break that off and then enter into that vapor or gas phase.
right. Okay.
Same principle behind, similar principle behind like if it's a solution. Um, okay. Weaker van der Waals forces.
Increased surface area, is that similar to increased surface tension?
Where do you see surface area?
I don't see it anywhere. I
Oh,
just
okay.
know that increased surface area leads to increased van der Waals forces.
Oh,
That's just in general, like it was something that came up.
well, Yeah, I'm not sure, but I know that surface tension would increase from greater overall greater intermolecular forces, which includes van der Waals forces,
Gotcha.
right? Because it's, the keeping the water molecules bound together. So C should be the opposite. Oh, now I see surface area for D. Um, Smaller droplets with small. Oh, it's okay. Droplet. Um, I see. Okay. Okay. So surface tension, surface area. My guess would be if you have a smaller surface area, you would be able to, like, if I just took this and I like cut it in half or something like that.
Yeah.
Smaller surface areas, um, water has greater surface tension. It creates droplets. Oh, okay. So kind of like this cohesive thing, like the greater the cohesive forces, the more spherical the droplet will be compared to a one with a lower surface tension, which is going to be more flat and more adhesive, like this one on the left. So, so okay, this one has high surface area, low surface area, but this would have low surface tension. This one should have high surface tension. Um, so yeah.
That doesn't make sense to me. Can you re-explain?
Okay. So we could have a bunch more like so we could have cohesive forces like all around here. Sorry, the surface tension forces would be like all throughout there. And if it's cohesive, like I would say like within it even, there's going to be a lot of bonds, intermolecular bonds with the water molecules themselves. Whereas if it was more flat like this, there could be, more of that surface tension only on like the outermost parts. So I think that the connection here that we have to make um is that this is that the same reason that this gives us this shape due to cohesive factors, right? Means that there will be a greater surface tension because you have water molecules bonding with other water molecules, and they can be very, very tightly and strongly bound, and that's cohesive forces, whereas opposite is adhesive forces, which is when like the water binds more to whatever container it's in or something like that. So yeah, if you have the droplets with smaller surface area, you're gonna have, oh yeah, they have a perfect picture here. So we can see here that we have our weak attraction here if you have your high surface area, whereas if you have a low surface area, it's more like round. And that's also due to the stronger attractive forces, which is what occurs from the cohesive force. Yeah, here we go. So thin elastic film. surface tension arises from cohesive forces between like molecules in a fluid. Water has a relatively high surface tension compared to many other liquids because ability to form hydrogen bonds. And then surface tension effects. So water molecules away from the liquid air interface experience intermolecular forces from all directions. Conversely, water molecules at the surface experience a net inward force. This decreases the surface area of the water and pulls it into an approximate spherical shape because the high surface tension indicates high cohesive forces. Water with greater surface tension creates droplets with smaller surface areas. So yeah, if you think about this droplet on the left, it has stronger adhesive forces because, for instance, this water molecule here is also forming bonds with whatever the surface is,
Yeah.
whereas high surface tension, you have a lot more of the water molecules bonding with itself, and that would give you that droplet. And that's also like why in space, uh, water droplets in space would be spheres. In fact, that's why everything, planets are spheres, stars are spheres. It's a way, it's the most minimal amount of surface area
Yeah.
if something is a sphere. So that's also a reason why that submersible like imploded because you're supposed to make them spherical. So you have an equal pressure everywhere. But he made it like this. Something like that.
Got it.
So yeah, so I would just take away from this that surface tension is caused by cohesive forces, which means that you have stronger bonds between, like if we're talking about water, between the water molecules themselves. Um, and surface area, right, is going to, so if you have more of those cohesive forces, it's going to want to bind to itself and, be more of a spherical shape, which means that we're having a smaller surface area. So I would say cohesive forces proportional to surface tension, inversely proportional to surface area.
Mhm. Got it. Yeah.
Did they even ask any questions about the math of the equations?
Yeah, they did, but I got it right.
Good. Nice, nice.
Okay. So yeah, I think I understand now. Um, service. So wait, just to confirm, cohesive forces are proportional to surface tension. Inverse proportional to surface area. So high surface area means lower surface tension, basically.
Hmm.
Yeah. All right. Okay. So that's it. Um,
Yeah. Here's a little thing here. So, surface tension is a type of force that acts to minimize the surface of liquids. Liquid molecules are pulling together with other molecules around them. This force is called a cohesive force. On the other hand, molecules on the surface have a cohesive force below the surface, but on the surface, they also have an adhesion that pulls them out with different substances. When the cohesive force is large and the adhesion like force is small, it can minimize the interface area to maximize the cohesive force. Then it says water has a high surface tension that can allow, as you said, like the water strider to float on water. dewdrops on the grass have a round shape, phenomenon that water does not spill easily, even if a clip or a coin is put into a, a glass filled with water. I'm not sure what that part means, but you can see that like if you hover over these molecules and you hover over the ones that are on the surface, you can see that it's showing bonds with the ones like, on the left of it, on the right of it, right?
Yeah, I understand.
And yeah, if you go towards the more inner ones, you can see that it's pulling in everything around it. So,
Okay.
and you could even like, pull one out and, have it be separate.
Okay.
Yeah,
Yeah, I'll go over this later.
sure, sure.
I just have to run, so
Yep, of course. So, let's see. Should be 163 for today.
okay.
All right, awesome. So, I guess you'll be continuing section bank and all of that stuff, and that's right
Yeah.
on Halloween. So
Yeah.
enjoy that and good luck. All right.
Thank you. I'll see you.